everybody, welcome to today's discussion video. I wanted to do a quick Spark Notes version of the entire kidney filtration cycle. I think a lot of people get bogged down in the details that we forget to connect what's happening at each step to the overall meaning of why is this happening, what is the body trying to accomplish with each of these steps. So I'm going to try to hit home the main takeaway messages from each step. So hopefully that will kind of solidify it in your brain what's occurring at each step and why, and make more conceptual sense. All right, so the first step when the blood is entering the kidney is the filtration step. So here we can see that there's the blood that's entering into the renal corpuscle. And what's happening at this stage is that there's filtration going on. So filtration is a type of passive transport that will essentially utilize some sort of pressure gradient to make something move across the membrane. So we can see here on this picture, on the membrane of the blood vessels, there are these little pores that are open and will allow basically anything that gets kind of pushed into it to cross out the other side. So the blood pressure that's flowing in is going to cause all of these solutes to be pushing up against this membrane. And so anything that can fit through those pores will. So this is a really non-specific mechanism of getting things to cross because it's literally just based on size. You can think of other modes of passive diffusion, like facilitated diffusion, for example. Those are going to be more specific because we have specific channel proteins that will bind to a certain substrate and allow those specific substrates in or out. But remember that with filtration, it's less specific because we're only based on size. So yeah, step one, key takeaways are that when that blood is pushing these solutes through these pores, everything that was in the blood that was carried in here is going to cross the apical side into the true urine, going to form that initial filtrate. And the only thing that was going to be left behind are large proteins that are too big to fit through. Okay, so step two, we go to the proximal tubule. Now in step two, essentially what we're trying to accomplish is all of the stuff that we just formed the pre-urine with, we want to harness a lot of that back because a lot of those things like salts, glucose, water, vitamins, blah, 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 those are all things that we actually need for other stuff. They're useful for us still. So it's actually probably going to be easier and selected for if we can find a way just to retain them and reuse them rather than excrete them and have to build them up from scratch again. So ultimately we are using this step as a way to kind of claim back what we want. Still, we obviously have waste that we want to excrete, so we're not going to pull everything back. So hopefully you can remember that urea is the waste product that we want to get rid of. Urea, it does not do us any good. We don't want to retain it for anything. So we do want that to be excreted. Everything else we're going to want to pull through back into the blood. So now I'm going to go through more of the mechanisms of how that happens. So here's a diagram of an epithelial cell. Just a reminder that this is going to be the basolateral side. So the bloodstream is going to be down here, and the pre-urine filtrate side is going to be up here. And so this whole reabsorption step is actually composed of several steps in and of itself. So I'm just going to go step by step for this part too, so that you can see what's happening and why. Because remember, our whole goal for this step is to get all of these solutes that we want back into the cell. The only way that we're actually going to be able to do that is by setting up a secondary active transport system. So hopefully if you can remember back to exam 1 content, setting up that concentration gradient is going to be key in allowing the next step to happen, which is what we care about. Okay, so with these pumps, you see that there's 3 sodiums being pushed out. There is 2 being pushed, potassium's being pushed in, but then it can also diffuse back out through this passive channel. So ultimately we're getting this net positive charge out here relative to in here. Because each time these pumps are moving, we're getting more and more positive charge out here, which is making this more and more negative. So essentially, that's the same thing as saying that we're setting up a concentration gradient. And so the next step is going to be the secondary part of secondary active transport, where we're using the passive, which hopefully you remember is when we're using sort of passive flow that's still going to allow us to do what we want. So we can look here, and now we're looking at these channels up here. So these are all symporters. And so remember that here we said that there's going to be a collection of negative charges because all of these positive charges are going to condense down here. And so all of the negative charges are going to be in here. And so the positive charges of these sodium ions are going to be attracted to the inside because remember, we're always trying to get to a state of more stability. And that's why this part is technically passive because we're doing something that's going with the flow of the gradient, with the flow of nature. And remember that when we're going towards a state of higher stability, that's going to be naturally an exergonic reaction. It's going to happen passively. And so we can utilize that energy, that exergonic energy, to also accomplish something else. And so in this case, we're going to be pulling along either glucose or these other ions or vitamins, all of that other stuff that we wanted to collect out here. We're able to couple that with this natural flow of the sodium ions into the cell as a way to essentially move these solutes that wouldn't be able to move on their own. So that's a really good example of secondary active transport because we're setting up the conditions that will allow this to happen even when we don't have active pumps that could move these solutes directly. Next, we just saw that these symports are going to be really, really active because we're trying to neutralize these charges. So we're going to have this influx of all of these solutes, which is going to cause all these solutes here. Now we have this really condensed hypertonic area relative to outside out of the bloodstream. And so what's going to happen is the solutes are going to want to move down gradient to neutralize and make this an isotonic environment again. Because remember, whenever we have hypertonic and hypotonic, we really want to neutralize that to get to an isotonic environment. And because we have these transporters that allow these solutes to move, that means that they can actually move out of the cell. If you remember the examples that we talked about, where only the water could move across the membrane, here, since we have those channels available, that's why this movement of the solute is actually allowed and not just the water. And once these are flowing out of this channel, remember that this is the bloodstream side. So this is the step that's ultimately what we wanted to achieve from the beginning, which is increasing the level in the bloodstream so that we can actually like send it along for other uses, rather than having it out here in the filtrate that's going to be excreted. And another interesting thing to note is that with these like solute movements, we're also able to like push water along with it. So I don't think I mentioned this step, but when we get this flow of all these solutes from the filtrate into the cell, we're now making this more hypertonic than this. So the water that's out here is going to be flowing in through aquaporins in order to help neutralize that. Because remember, we're always trying to get to an isotonic state. So water is also going to flow through at this point. And then again, when we have all of these solutes that are now moving here, that will eventually cause this, which started off really hypertonic, to actually be hypotonic, because these are all going to be moving out here instead. And so again, the water is going to be like, wait up and just try to catch up with those solutes and neutralize that into an isotonic environment. So again, we're getting the water flowing through the aquaporins at that stage. So ultimately, this was the same slide from before, but we could see that, so we have just the urea in the pre-urine at this point. We do have trace amounts of like protein and some other stuff, but like the whole goal was for urea, and that's going to be like the majority of the contents. But now in the bloodstream, we have those large proteins. Those were there from the beginning because remember, they couldn't fit through the porins in the renal corpuscle, so those have been there. But now we have salts and water and glucose and vitamins and amino acids. All those things that we wanted to keep and not pass on through the excretion cycle are going to be back in the bloodstream. So that's really good. And an important thing to remember is that so we're able to like reabsorb about 2/3 of the original salt content and water content. And so one thing to remember is like when we're moving equal amounts of salt and water, that's going to make the concentration of those things be the same. So even though we have less of like the actual like absolute amounts in the filtrate, the concentration is staying the same. Okay, so after this point, now we're going to move on to the loop of Henle. The advantage of the loop of Henle is setting up this like differential permeability state so that we can use this like interesting structure, like highly absorb what we want to absorb. And this is a mechanism of positive regulation. So this side of the loop of Henle is going to have a bunch of aquaporins lining on the basal side of the epithelium. And remember that with aquaporins, you can move in either direction, like water can move either direction, but it's going to always move to whatever is more hypertonic so that it can like neutralize and make a more isotonic environment, which is more stable. And so in this case, this outer environment is going to be more hypertonic relative to inside the kidney. So the water is going to flow out and be recuperated into the bloodstream, which is ultimately like what we want because we don't want to like waste a bunch of water if we don't need to. And it also is really helpful because as we continue descending and we finally reach to this ascending loop, having all that water being pushed out now made this inner part like really hypertonic. So on the edges of the epithelial cells here, the basal edges, instead of having aquaporins, now we have these sodium pumps. And we have an environment in here that's actually like really, really concentrated with sodium because we just like essentially made all that water leave. So now, so that's like how this is a mechanism of positive regulation. And so now we have these pumps that are able to actively pump out the sodium ions because now it's just like trying to regulate again. It's like constantly trying to like play catch up of maintaining this isotonic environment. So the sodium is going to move outwards. And the reason here why we couldn't just like neutralize this by having the water move back in is because we don't have those aquaporins. We only have this. So only the electrolytes are going to be able to move. This step, essentially what we've done is we've changed like the relative concentrations. So since we moved a lot of the water and the sodium out, the relative proportion of urea at this point is really, really high. So that's ultimately serving our goal because remember, we want to get rid of the urea and hold on tightly to everything else. And so that is like a huge booster in this step. So next step is further water recovery in cases where we need it. So this part is taking place in the distal tubules and collecting duct, which occurs right after the loop of Henle. And so this is another example of regulation. So when we are in a dehydrated state, we're going to naturally produce antidiuretic hormone, and are also called ADH. And what ADH does, it recruits extra aquaporins into the collecting duct itself. So we can like see that there's going to be more aquaporins on these edges when we have this ADH recruiting them. And what that means is to flow out into and be like reclaimed into the bloodstream. So having these aquaporins, and we have this hypertonic environment out here. So remember, in the previous step, in the loop of Henle, we had just pumped out all the sodium ions. So it's going to be relatively hypertonic compared to the inside. And then inserting these aquaporins is going to allow for that movement of the water out into the bloodstream to try to neutralize that. And that's good for cases of dehydration because that means that the water is going to stay in your body rather than coming out in your urine. So that's why this is a good example of regulation and adaptation. And this also explains why we have this like interesting relationship where the more hydrated you are, the clearer your urine is. Because we can see here, if you have this ADH that's stimulating all this water to be released, that's essentially making this inner part super concentrated with solute. So that's going to be like a darker yellow color. Whereas here, this is what's happening normally when you're not in a dehydrated state. Water is going to be able to flow through just fine because you already have enough. So there's no reason for more water to be absorbed. So this is going to be a clearer state So essentially, if you have really, really dark urine, you should probably drink more water. Right. And one last method of regulation to know for the kidneys is with blood pressure. So this is also occurring in the distal tubules and collecting duct. A low blood pressure state will stimulate the production of aldosterone. So when we have aldosterone being produced, that's going to stimulate sodium pumps to be added to the basal side of the epithelium. So remember before, when we were talking about ADH, we're having aquaporins added. And in this case, for blood pressure, we're adding extra sodium pumps. And why that's helpful is that allows active transport of sodium out into the bloodstream. And remember, that can happen because with active transport, you can go against the gradient. So even though we already have a hypertonic environment on the outside, since we have those pumps, we're able to push those out. And that causes the cells to become even more hypertonic relative to the inside. When we have that active transport of those extra sodium ions out into the bloodstream, that's going to cause like the renal tube to be even more hypotonic relative to the bloodstream. So that is going to stimulate water flow, which is going to ultimately increase blood volume and therefore blood pressure. All right, so that basically sums it up for the big picture SparkNotes version of the filtration system of our kidneys. I did want to go over some problem roulette questions. Okay, so this one says, which of the following is true of the loop of Henle? The osmolarity of the pre-urine is constant as it moves through the loop. The descending loop has epithelial cells with aquaporins in the apical membrane. The ascending loop has epithelial cells with aquaporins in the apical membrane. ADH controls how much water leaves the pre-urine in the loop of Henle, or none of the above. So I would take a second to pause and try to think about this before you move on. All right, and we can see that the answer is C. The descending loop has epithelial cells with aquaporins in the apical membrane. This is kind of just like you have to just like know this, but if you can remember what the structure looks like, remember that when we're descending, the first thing that's happening is having the water flow out. So we need those aquaporins here. And then it's not until we get to this ascending part where we have the sodium ions move. So that's kind of just like a know it or you don't type of thing. Okay, let's look at this next one. Which regions of the nephron allow for reabsorption of sodium from urine into the body? Renal corpuscle and collecting duct, proximal tubule and ascending limb of Henle, proximal tubule and descending limb of Henle, glomerulus and ascending limb of Henle, ascending limb of Henle, and collecting duct. So again, I'm going to give you guys a second to pause and try to think about this. All right, and the correct answer is B, the proximal tubule and ascending limb of the loop of Henle. So one big hint is that whenever you're talking about reabsorption steps of the nephron, proximal tubule is probably going to be the right answer because that's going to be a really important step in getting basically every single solute besides the urea back into the cell, and then ultimately the bloodstream. So that's going to, like, anytime you see reabsorption, like, that's going to kind of indicate proximal tubule. And then the reason why ascending limb is the second choice is because of this same structure that we just talked about here, where we're using these pumps to make all of the sodium ions travel into the bloodstream, which is what the question is asking about. Cool. All right. Inhibiting the sodium potassium pump in the proximal tubule of the nephron would lead to either a high concentration of urea in the urine, a small volume of concentrated urine, a lower concentration of sodium in the urine, a higher concentration of chlorine in the blood, or a higher concentration of glucose in the urine. So if you don't have this initial pump working, remember that this pump was the thing that was like setting up that gradient that allowed the secondary active transport of all the solutes that were in the filtrate to move into the cell and then into the bloodstream. So like inhibiting this first step is like automatically going to tell you that the glucose is going to be higher in the urine because there's going to be no movement into the cell. So it's all going to just like remain stagnant outside in the filtrate. Okay, individuals with diabetes do not produce ADH or have ADH receptors that cannot bind them. So which of the following is true? A, they produce a large volume of dilute urine. B, they produce urine that contains a lot of glucose. C, they produce a large volume of concentrated urine. D, they produce a small volume of concentrated urine. Or E, their collecting ducts contain many aquaporins. So again, I'll give you guys a second to pause. Alrighty, and we can see that the answer is actually A. They produce a large volume of dilute urine. So if we look back at the structure here, this is easy to see. So these are the two situations that we could have. So if we have this normally functioning system of ADH regulation, where having a dehydrated state will induce this ADH to increase, which will allow these aquaporins to be recruited, which will then allow the water to go out to reclaim more of that water for the actual use of the body rather than to excrete it. Now what we're saying is if we mutate something about this ADH system, so either the receptors or we don't even make ADH at all, something will happen where even when we have a dehydrated state, ultimately probably should increase the amount of water in the bloodstream, they're not going to be able to do that because they don't have that signal that's going to recruit those extra aquaporins. So at that point, you're going to get the same result as if you didn't have that dehydrated state. So that's like why it's a health issue, because we obviously need this form of regulation to help prevent like really bad dehydration. And that's also why hopefully that makes sense with the answer. So you're going to produce a large volume of dilute urine regardless of whether or not you're dehydrated. This question, which of the following is not affected by the sodium proton pump in the proximal tubule? Water, chlorine, sodium, glucose, or none of the above? And I'll give you a second to think. And the right answer was E, none of the options, because hopefully you can remember from this original structure. Okay, so we have this step of first we're setting up this gradient, which is going to push all of these things into the cell. And having this movement of the solute is also going to affect water movement. So even the water is moving via a different mechanism, it's still being affected ultimately by the actions of this pump. Because if we didn't have this pump, then we wouldn't have that movement. So we wouldn't have this like relative hypertonicity here. So the water wouldn't move. Even though it's like one step removed, it is still ultimately being affected by this pump. And then obviously with the next step, we have solutes again moving out into the bloodstream, and then the water again being kind of led by those solutes. I think like probably a trick answer or like a convincing answer might be water, because you might remember like, oh, there were those channels, like the secondary active transport channels were the ones for like all the solutes and stuff. But remember that the water is being affected because it's like kind of chasing where the solutes are going. So that is still going to be an answer. Okay, sweet. So this is actually the end of my discussion video. So I am going to actually make another review video going over the discussion exercise, because I want you guys to be sure that you understand like the concepts and how to do it. But I want to give you guys a really good shot of trying it on your own first. So I'm not going to tempt you with putting the answers inside the video. So yeah, this is going to be it for my discussion concept review, but make sure to keep your eyes peeled for that exercise review. And overall, this is going to be our last like asynchronous discussion, guys. This semester has gone by really fast, and I have really loved working with you guys, despite not actually like meeting you in person. I feel like being able to like meet you guys in office hours and over email and just in other like interesting ways has been really fun, and I'm glad that we have been able to kind of make this work. I know it's been kind of a challenge, and it's definitely been a learning curve. So I really appreciate you guys sticking with me through the semester as I kind of figured out like what structure works best and how to kind of help you guys be successful. Yeah, ultimately, I'm just like really proud of you guys, and I think that you are rock stars for just like really adapting to all of the challenges of the semester. Spring courses are hard in general. Spring science courses are particularly hard, and then spring science courses with this additional challenge of changing our structure has been really, really difficult, and I think that you guys have handled it really well. So again, I just wanted to thank you for your hard work and for everything. Once we kind of move back to traditional classes, if you guys ever run into me at the USB or like anywhere, even though I might not recognize you because a lot of you don't have your videos on when you come into discussion, please feel free to stop me and say hi. I would love to chat with you guys. And also I'm happy to chat about anything like unrelated to this course or different science majors here or career paths or anything like that. Feel free to reach out to me. I am definitely an open resource for you. And also I'm teaching microbiome for the summer term. So if you're taking microbiome, which is like bio 207, you're taking that summer term and you will not be rid of me. So now's your chance to drop the class if you want. But anyway, thank you guys, and I will see you for my later discussion exercise video. Bye.